Hello, everyone. 1115 wants us to know how we use antigens to determine blood type. Now, this might seem counterintuitive or maybe a little bit backwards, but we call the proteins on the surface of our red blood cells antigens. Oh, that's weird, Mr. Fleming. I thought antigens were non self. It'll make sense once we talk about how we use this thing called monoclonal antibodies to determine blood type. But just for review, blood group B has B antigens on them. Blood group AB has both, right? It has both of them on there. And then if you have blood type 0, you don't have any antigens on. So let's talk about why we use this term antigen for a protein that's on our red blood cells in this process of making monoclonal antibodies. All right, so I thought there was a way to clear this. Okay, so describe the production of monoclonal antibodies and their use in diagnosis and treatment, but also determining blood type, which I guess is kind of like a diagnosis. Mono means one, clone means clones. So what we're doing is we're going to have one cell that we've cloned make the antibodies for us. All right, so let's do this. So we take this poor mouse and we inject it with a human blood type, right? If I put blood type A in this mouse, the blood is going to say this, or the mouse's immune system is going to say, hey, this is non self. This isn't me. I need to produce antibodies for this. And the mouse will have an immune response to that human's red blood cell. Now, remember, plasma cells, we have B cells that can be memory cells or plasma cells. And plasma cells are the ones that make the antibodies. So the mouse is going to start this whole clonal selection process that we talked about to make antibodies specific against blood type A. Now, there's two challenges here. One is we want the cells to stay around for a while, and we want them to reproduce and keep dividing and keep dividing. And the best way to do that is to take a tumor cell, right? If we can take a cancer cell, because that's what cancer is. It just just divides uncontrollably. So we actually stick them together. We fuse them together into um, one cell. So now we've got this antibody A, right? It's making antibodies against blood type A. This is our antigen, and it's a machine, and it just keeps dividing and dividing and dividing, and making more and more of this. So let's pretend for a second that I am blood type A. And how they determine my blood type is they will take blood from my body, put it like in a little test tube. Here's all my red blood cells. And if they add these antibodies, right? These are little antibodies. If they add that in there, these are going to start sticking to all my red blood cells. So we have antibodies sticking to them, and they're going to stick to other antibodies because that's what antibodies do. They stick to each other, and they just like are going to connect all these red blood cells into a gigantic clump. So if they add that to my blood, you'll start noticing there's a whole bunch of blood cells stuck together here, a whole bunch of blood cells stuck together here. And it's not going to be this solid red color. It'll be all like clumpy, like old milk. Like it's called a agglutinated red blood cells. And that's how we use um, this monoclonal production of antibodies to determine blood type.